Previously, we discussed about water imbibition as the first key step in germination, as well as the different factors affecting the rate of imbibition in dry mature seeds. In this lecture, we will cover the second step in germination, which is enzyme activation. Specifically, we will look at how the different storage reserves in seeds are broken down to provide food for the growing embryo during germination, beginning with carbohydrate metabolism. Recall that seeds take in water at three different phases during the entire process of germination. Phase 1, as you can see here, is defined by a rapid water uptake by the seed. This phase is also known as the imbibition phase. When the seed takes in enough water to reactivate its metabolic activities, water uptake gradually levels off, as you can see in this part of the graph. This is followed by another increase in water uptake at phase 3, when the radicle or the embryonic root emerges and aids in water uptake. Now, sometime between phase 1 and phase 2, enzyme activation occurs. Enzymes are proteins that act as catalysts for chemical reactions. In germinating seeds, enzyme activation serves to break down stored reserves, aid in the transfer of nutrients from the storage reserves to the growing embryo, and trigger chemical reactions that use the broken down products for the synthesis of new materials. All these metabolic processes require water, and that is the reason why we see a leveling off of water uptake in this stage following enzyme activation. This is not because the seed stopped taking in water for a few hours. It is because whatever water that the seed is uptaking is being used for the metabolic processes that were reactivated during the second phase of water uptake. Enzymes that break down carbohydrates, proteins, lipids, and minerals are the first to be activated between phase 1 and 2 of water uptake. To provide nourishment or food to the growing embryo, the storage reserves must first be broken down to soluble forms. The broken down products are then transferred from the endosperm to the embryo. Only then can the products be transformed into energy molecules that can be immediately used by the growing embryo. We will now go through the general process of how each of the storage reserves in the seeds are broken down, starting with carbohydrates. You already know from previous lectures that starch is the major form of carbohydrates in seeds. Starch can be of two types, amylose and amylopectin. You can see the structures of both starches in this panel. Amylose has a linear structure, while amylopectin has a branch structure. Both starches are composed of glucose units, which are represented by each of these structures that you see here. For the seed to be able to use up the starch as food, they need to be broken down first into its simplest component, which is glucose. This is where enzymes come in. There are two classes of enzymes that can break down starch into smaller components, the phosphorylated enzymes and the hydrolytic enzymes. The phosphorylated enzymes break starch in the presence of inorganic phosphate. That is why they are called phosphorylated, because their activity requires phosphate. The hydrolytic enzymes, on the other hand, break up starch in the presence of water. That is why they are called hydrolytic, from the prefix hydro, meaning water, because their activity requires water. There are three enzymes involved in the phosphorylated degradation of starch. We have starch phosphorylase, the branching enzyme, and glucosyltransferase. Again, all these enzymes fall under the phosphorylated class of enzymes, meaning they require inorganic phosphate to be active. Let's take a look at how each of these enzymes degrade starch in the form of amylopectin. Recall that amylopectin, like your amylose, is a form of starch composed of glucose units. Each of these structures that you see here is one glucose unit. What starch phosphorylase does is cut individual glucose residues from the non-reducing end of the starch molecule to generate glucose 1-phosphate. Starch phosphorylase can only break down bonds located at least 4 glucose residues from a branch point. In this example, our branch point is right here. So we have to move 4 glucose molecules up. 1, 2, 3, and 4. So imagine another glucose molecule here. That imaginary glucose molecule is where starch phosphorylase should stop cutting because it can only break down bonds located 4 glucose units from a branch point. What will happen to the remaining parts of the amylopectin that cannot be degraded by starch phosphorylase? This is where the debranching enzyme or the polulanase or the R enzyme comes in. From the name of the enzyme itself, we can deduce that the debranching enzyme cuts through the branches in the amylopectin to release small linear chains of glucose. Once these smaller chains are released, starch phosphorylase can start cutting them down into single glucose units. Another enzyme, known as the glucosyltransferase, puts together the smaller chains together so that it will be easier for the starch phosphorylase to break them down. This is an overview of how the three phosphorylated enzymes break down amylopectin. We have here our amylopectin, which is a form of starch. During phase 2 of water uptake, starch phosphorylase starts cutting through the amylopectin one glucose at a time. But it cannot cut through the branches in the amylopectin, so smaller but branched amylopectin starts accumulating. The debranching enzyme comes in to cut those branches. Without the branches, what we have are essentially amylose, right? So starch phosphorylase continues to degrade these shorter linear glucose chains. Alongside that, the glucosyltransferase stitches together the smaller glucose chains for the starch phosphorylase to cut. The final products of this reaction are glucose units, which are converted into glucose 6-phosphate. Those are just phosphorylated enzymes. Remember that there is another set of enzymes that I mentioned earlier that can degrade starch, and these are known as hydrolytic enzymes. Again, these enzymes require water for their activity, and that is why they are called hydrolytic enzymes. What are some of these hydrolytic enzymes? We have alpha amylase, beta amylase, and alpha glucosidase. Let's now take a look at how these hydrolytic enzymes break down starch using amylose as an example. Amylose is just a linear chain of glucose units. In breaking down amylose, the objective is to cut it down into its simplest unit, which is the glucose molecule. Let's start with alpha amylases. The alpha amylase cut into the amylose chain to make essentially shorter chains of glucose. But the smallest unit that alpha amylase digestion can produce is a maltose, which is essentially a 2-glucose molecule. We also have beta amylase. Beta amylase can cut into amylose one maltose residue at a time. Maltose, as I said earlier, is composed of 2 glucose molecules. So we can see in this diagram our beta amylase here and here. And you can see that whenever it cuts through amylose or the shorter chain amylose, it produces maltose residues. But maltose is still composed of 2 glucose units. It still needs to be broken down into individual glucose units so the seed can use it as an energy source. This is where alpha glucosidase comes in. Alpha glucosidase can cut into maltose residues to produce 2 individual glucose units. The alpha glucosidase can also cleave or cut the shorter glucose chains produced by alpha amylase into single glucose molecule. However, it cannot directly attack the full amylose molecule. This figure here provides an overview of how hydrolytic enzymes degrade amylose. Amylose can be a substrate for both alpha amylase and beta amylase. The alpha amylase degrades amylose into shorter chain glucans, while beta amylase cuts through the amylose one maltose residue at a time. Now, the shorter glucose chain produced by the alpha amylase can further be degraded into smaller residues by alpha amylase, beta amylase, and alpha glucosidase. The products of both alpha and beta amylase is maltose, which is still a 2-glucose molecule. To break the maltose down to single glucose units, alpha glucosidase comes in to do the job. Alpha glucosidase can also cut the shorter glucose chains produced by alpha amylase to directly produce single glucose molecules. Amylopectin can also be degraded by the hydrolytic enzymes. The only difference is that in addition to the hydrolytic enzymes, breakdown of amylopectin also requires the presence of the debranching enzyme, which will cut the branches of glucose chains in amylopectin. In monocots, enzyme activation is regulated by plant hormones. Plant hormones are substances that are present in minute traces in the plant. They are responsible for regulating plant growth and development, as well as the plant's responses to external environments. One of the key hormones involved in seed germination is known as gibberellic acid or gibberellin. In a monocot seed like any of your cereal grains, gibberellin mediates the activation of enzymes necessary for the breakdown of stored reserves, particularly of starches. This figure illustrates the process of gibberellin-mediated enzyme activation in barley seeds. When the seed has taken in enough water, maybe around 50% of its original dry weight, gibberellin is released from the embryo axis and scutellum into the allurone layer. The allurone layer, if you recall, is active in the seeds, and it can synthesize large amounts of enzymes. When the allurone receives the gibberellin, it is induced to synthesize enzymes that can degrade storage carbohydrates and proteins. These enzymes include proteases, amylases, and glucosidase. These newly synthesized enzymes are then transported to the starch endosperm, where it starts degrading the starch. The final product of starch degradation is sugar in the form of glucose. These sugars are absorbed by the scutellum for transport to the developing embryo axis. In summary, stored reserves in the form of carbohydrates, proteins, lipids, and minerals need to be first broken down into smaller soluble subunits, translocated to the growing embryo, and transformed to energy molecules before it can be used by the embryo during germination. The enzymes that break down the storage reserves in the seeds are activated between phase 1 and 2 of water uptake. Particularly in monocots, the activation of enzymes is mediated by the plant hormone gibberellic acid. Stored carbohydrates in seeds are degraded via two pathways, the phosphorylated and hydrolytic pathways. Enzymes involved in the phosphorylated degradation of carbohydrates include starch phosphorylase, the branching enzyme, and glucosyltransferase. This pathway requires the presence of inorganic phosphate to break down starch. Enzymes involved in the hydrolytic degradation of carbohydrates include alpha and beta amylase and beta glucosidase. This pathway requires the presence of water to break down starch.